right, I'm gonna use the since Alan brought it, provided by Matt. I'm calling to order the 1245th regular meeting of the Entomological Society of. And as a reminder, this is our last regular spring meeting before our membership potluck banquet on June 8th and the resumption of regular meetings in September. A reminder for those of you in attendance, Venmo or cash for your pizza slices. What was the estimate down on that for you, Matt? Like some $2, $2 a slice. That's a bargain, actually. There's still a little bruschetta and some other beverages over there. Okay. I'm gonna first call on Secretary Gary Hubble to read our approval of the minutes of last meeting.
At 7 p.m. on April the 4th, 2024, the regular meeting of the Entomological Society of Washington was called to order by President Ron Weber in room WG 33 of the National Museum of Natural History. Present for the meeting were 13 members, 8 guests, and 15 virtual attendees. Recording Secretary Gary Hubble read the minutes of the March meeting, and these were approved by attending members. President Weber offered a few updates of society business. The adjusted bylaws are currently being reviewed by a lawyer and thus not available for presentation. The U.S. Postal Service requires changes in the cover of the proceedings of the ESW, and this is being undertaken. The annual banquet will be held at the Lincoln Lodge in Beltsville on the 8th of June, and shuttles will be available to that venue. Curator Talita Simone has requested assistance in the movement and placement of society publications. Lourdes Chamorro has received ESW mugs and virtually displayed one. Past President Matt Buffington commented that the mugs are of good quality but need a logo adjustment. The mugs will be available on the ESW website probably by early May. Membership Officer Elizabeth Young virtually noted that there are 3 new applicants for society membership: Jeremy Frank, Lance Jones, and Y. Turner. Program Chair Alan Norbaum mentioned that the speaker for the May meeting will be Karin Burghardt, then introduced the speaker for the evening, Rebecca, who is currently a postdoc worker at the Museum of Natural History. The title of the presentation was Superbugs, Aphids Taking Over the World. Rebecca received her doctorate degree in 2022 and had studied termites. She has been working with Chris Owens at the National Museum on aphids. Aphids are quite important to crops. They carry plant viruses and have relationships with bacteria. Over 5,000 species of aphids have been described, and of these, some 450 species have colonized food and fiber crops. As direct feeders on plant sap, they damage crops and reduce yields, but have a greater impact as vectors of plant viruses. In the United States, 430 invasive species cause billions of dollars of agricultural damage. Analyses of hybridization have yielded many important findings. Aphids reproduce rapidly and young are born pregnant. Increased research in genetics of aphids continues to aid control. After her presentation, Rebecca surprised the audience by singing 2 songs about insects, accompanying herself on a guitar.
It's gonna be hard.
The original songs were educational, and one involved an audience response sing-along with lyrics provided on the screen. Program Chair Alan Norbaum commented that the musical surprise was likely unprecedented in the history of the society. Everyone was delighted with the sub-event. The meeting was adjourned at 8:15 p.m. Corrections, yes.
Minor correction, the building that the banquet will take place in is known as the Log Lodge.
Oh, okay.
I mean, there are Lincoln logs.
Yeah, yeah.
That's another thing.
Log Lodge, okay, thank you.
It's built by the CCC.
My bad. Email?
All right. Everybody okay with the minutes? All those approving of minutes, please say aye.
Aye.
Any objections, like nays, abstentions? So ordered. Okay. We have the report of officers and committees, which I'll keep brief. And say that the draft of the revised bylaws is not surprisingly still with a lawyer, but thanks you to Abby for all her work on this. We are that close, Andrew? We're this close to having a speaker for the banquet, and Andrew's done all the logistics for the Saturday, June 8th meeting at the Log Lodge, which is on the Belso Agricultural Research Center campus. And as mentioned before, we'll provide shuttle from the Greenbelt Metro. We're this close, don't worry, we'll have a good speaker. We had a little hiccup when people say no at the last minute, but it'll happen. I want to report from Elizabeth Young, our Membership and Communication Secretary, 3 new members. Yavuz Turan, who is Assistant Professor at the Department of Biology at Marmara University in Turkey, whose interests include the step biology. Lance Jones, who's a PhD student in Stephen Downey's lab at the University of Illinois, Urbana-Champaign, who's studying the Depressariidae, Weber moths, and previously studied with David Grimaldi on Drosophila at AMH in New York. And finally, Jeremy Sloan, who's currently with Bartlett Tree Experts and recently completed a PhD in entomology from NC State. Margarita Lopez Uribe at Penn State on honeybees and integrated pollinator pest control. That's new members. And I want to ask if we have any other officer reports or other unfinished ones. And then comes show and tell. Alan has at least one show and tell. You have to bring your show and tell, shows to tell about. Okay.
So I was out collecting yesterday. One thing I was looking for was there's a leaf mining tephritid that gets into Pecara aurea. And somebody gave me a tip when they saw some leaf mines. So they're actually not, they're tephritid. I think they might be something your alley, Don. But if anybody wants to look at them, anybody wants them.
What's the plant again?
Pecara aurea, which is the
Oh, yeah.
So it's like a
So
single-
golden ragwort.
yeah, it's nice. I think the top 2 leaves still have insects.
What's the leaf miner that you got all winter amongst those guys? Like I have pictures like from January, February in snow where they're still like actively mining. You can see it's heat. So I think they overwinter as part. Okay, cool.
I don't think I tend to know the tephridids.
Right, yeah.
And there's a
No,
genus.
normal. That's the stuff we
It
stuff.
makes a big.
Valley of Grass Lorraine.
This is more.
Smaller than the gymnastic.
Yeah, this one's like. I think it's very good. And then there's a genus called Straußia. We had a speaker 3 years ago talk about it. They're stem miners on various composites, especially sunflower. And there's 3 species that are on Helianthus tuberosus, including one that has like well, the females usually have these banded wings, and then the males of some species look like the females, but others have this sort of coalesce pattern fused together. Anyway, Helianthus tuberosus has 3 different species on it, one of which has this kind of coalesce pattern. There's another species around here, Helianthus decapetalus, And when I was working in the Smokies, we found one definite species on that, and then there were a couple of males that came out that also had this kind of coalesced patterns. Anyway, I was looking on the plant here, and yesterday there were a whole bunch of males.
I can't tell. Not
There's a pair in there.
Elianthus tuberose is also known as Jerusalem artichoke or sunchoke. For the non-virus. And were they lecking, Alan, or what? Don't they leck?
I wasn't looking carefully enough to tell.
Okay.
I was expecting probably to find the other thing. I didn't find any of them. Probably previously unknown species.
What would you look for if we were going to look for them
Yeah.
on that other species? It's a mine,
I don't
but it's
know.
not a blotch mine, or is it?
Yeah, so the adults are out now, and the larvae mine
Diptera?
stem
Oh,
over the summer,
stem.
so they're not mature until.
Would you look for a wilted stem or you see? It's pretty much there to
They're
come.
pretty much in the pith. I don't think they really damaged anybody. So
Later.
late in the fall, you cut the stem and you can usually see the evidence in the line. And then they burrow down into the
Don't you think?
such best places on. Sure.
Okay, do we have any other exhibits or show tell, or anyone online want to make some show and tell such as it would be possible virtually? Alan, anybody there? Okay. Well, if we have no further notes or exhibition of specimens, I'll turn it over to Alan to introduce our evening speaker.
Okay, thanks, Don. And Don gets credit for arranging this presentation. And our speaker tonight is Dr. Karin Burghardt, who's an assistant professor in entomology at the University of Maryland College Park. And she's also a research associate at the Smithsonian Environmental Research Center. Trained as a community ecologist, she specializes in understanding plant-insect interactions in human-modified landscapes, birds, abandoned agricultural fields, managed forests. By examining how human management practices alter support the biodiversity of these spaces, the lab's research program helps determine best practices for how humans can share space with a variety of flora and fauna. Karin received her PhD in ecology and evolutionary biology from Yale University, working with Oswald Schmidt on herbivore and fences and goldenrod plants. Prior to that, she received her BS from Delaware in 2007, working with Doug Talmy, role of native plants ecosystem.
Going to try to escape from behind the curtain. We'll see how it goes. All right, so thank you, Alan, for the introduction, and Dawn to speak to everybody today. I'm Karin Burghardt. We were talking a little bit about this caterpillar. I like caterpillars a lot, and we love caterpillar pictures. And I feel like this is the Entomological Society, so you have to put up with it. I feel like sometimes I don't go as much into detail on individual insects because my general are not as interested. But this guy, the morning glory prominence, and it's kind of, and you can sort of tell because it's the pomone, it's the specific name, but it actually doesn't feed on morning glory. It's kind of a misnomer, and so Wagner in his book has started calling this the checkered fringe prominence rather than the morning glory. We find it in the experiment today at SERC, primarily feeding on oak, but it is actually a fairly generalized feeder across a number of species. You leave it for like a few minutes and then we might just have to click. Yeah, we just have to like click the screen once. Okay, so where am I coming from on this? So I am not a systematist. I'm going to feel a little bit like an interloper here. Instead, I'm an ecologist and I'm really interactions. Is that coming from up there?
Yeah.
Someone in the meeting.
Somebody in the meeting. Could you maybe mute everybody from your side? But I started getting interested in insects when I was an undergrad and I was working with Doug Tallamy, and we were collecting all of these insects off of native and non-native plants. And then the recession hit. It was 2007, and I couldn't get a job anywhere. Like Doug took pity on me and gave me a job like identifying all these insects we collect. And so spending years just looking at insects under a microscope and identifying them, I started just getting really interested in how all of these insects were interacting with the plant. So these creatures have figured out ways of exploiting virtually all of the different parts of the tissue on the plant. So you have insects that are feeding on the main leaf tissue by chewing, like on the previous picture, but then you've got all these miners that are feeding inside the leaves. And you have insects that have figured out how to feed on really highly pressurized xylem tissue, which is crazy that they figured out how to do this. So this is sort of how I came into this. And I really spent most of my time growing up in the Smoky Mountains, but then moved to Delaware when I went to undergrad. And so since then, really my home base has been the Mid-Atlantic. So the insects that I like to know about and I like to work with are kind of in this range along the coastal Mid-Atlantic. And I came back to the DC area, or came to the DC area about 9 years ago when I started doing a postdoc at the Smithsonian Department of Entomology. So I worked there for 2 years before starting at Maryland as assistant professor. And so that's the work I'm going to be talking about primarily today, is work that I started when I moved to start a postdoc at the Smithsonian. And I think that working with creatures that are local to the area where you're teaching is really powerful for students because they can go out and find these creatures. So the work my lab does is on plant-insect interactions, but it's really about how human modification of environments alters plant-insect interactions. And so then if we know how human management is kind of changing these interactions, that provides opportunities for us to maybe tweak our practices or figure out ways of, in particular, what we'd be talking about as plants, kind of rearranging things on the landscape so that we can kind of maximize diversity and make sure that our ecosystems have lots of different players. And so this is the broad theme of the lab. But today, I'm going to really be focused in on one piece here, which is really this expansion. So the lab does work in suburban yards and in urban areas and street trees. But this particular experiment is an experiment on tree diversity that I started working in 2017. And I think we're finally figuring out some answers from now. How many years later? So I'm going to let you in on a little bit of that. So the whole theme is kind of going to be about this idea of, does it matter what the diversity of trees around you are if you're a tree? And this is from the tree perspective, but it also from the perspective of all of the creatures that are going to use these individual trees. And so I'm going to start talking primarily about herbivores, which is what I really like to think about. And I'm going to give you 2 vignettes, one about caterpillars and one about cicadas. Cicada work is actually Kristen's master's thesis work that she just defended successfully last fall. And then we're going to kind of think about, okay, well, how does tree diversity matter for these herbivores and how they use those plants? Then how does diversity change the way plants are actually functioning on the landscape in terms of their growth and their traits? And then lastly, we're going to kind of jump up to predators. And I'm kind of creating an herbivore sandwich here. where we're going to go from the middle out. So we're going to talk about the herbivores in the middle, we're going to go to the plants, then we're going to go to the predators, and then try to see if there's some lessons that we can pull from it about overall how planting diversity alters the interactions of critters on plants. So one of the reasons I'm really interested in this question of diversity, and particularly tree diversity, is that humans plant trees kind of everywhere. This is like something we do, especially on the East Coast, where trees, where it's a temperate ecosystem and trees are a native part of the habitat. But we're also doing this out in, in Utah and in places where actually trees don't tend to natively exist in habitats, but humans like trees. So we plant them everywhere. Often we do this as single species. So there's a development that just went in down the street from my house, and it is virtually all red maples. Like huge development, they went in and put all red maples. Red maple is a great tree, nothing against red maple. But that is one way that we have we can decrease diversity, but also we grow a lot of tree plantations that are often single species. And this is making up a pretty large proportion of the world's forest area, about 7% right now. We also plant trees for other reasons. So we can plant trees to restock forests after we harvest. We are doing a ton of restoration work now where we're increasing the number of trees along, say, riparian buffers and other areas. We are using it in cities for urban cooling. So another sort of part of the lab's work is all focused on street trees and some of the services those provide and the pests that are on them. And urban cooling is a major service along with stormwater and cities. So we're putting a lot of trees out there on the landscape that didn't used to be there. And one of the questions, and so for example, in Maryland, we're about to plant 5 million trees in the next 10 years. This is a bill that passed in 2001, so we got like 8 years left to plant a number of trees. Pennsylvania has one of these too. DC, I guess, probably doesn't have that much land. I don't know if Virginia has one of these programs, but we're doing a lot of this. And so my question is, as we're kind of going and putting all these trees out in the landscape for carbon storage, for all of these different projects, does it matter how we plant them for all of the critters that are going to use these? Because these plants could also be sort of providing the baseline food source for food webs that are going to develop in these human landscapes. Insects are going to feed on those trees. Birds are going to come and feed on those insects. In this tree experiment, I'm not going to talk about, but there's actually lots of little mice and things that crawl up and will attack our like plastic caterpillars that are on there. So there's all kinds of food webs that are built just by putting trees. And so if you're kind of a moth that is, my pointers are working, but if you're a moth like this one, sort of flying around in a forest structure, do you interact with that forest differently when it is all the same species versus if it is multiple different species, where you're having to potentially find your host in a sea of other hosts, and you're having to differentiate those guys from one another? And so this motivated this experiment that I'm going to be spending my time talking about for the rest of the evening. This is a large-scale tree diversity experiment that was basically planted to answer this question of how does tree planting diversity affect how a forest grows? And so this is at SERC. This is in Edgewater, Maryland, just south of Annapolis. You all have been over there. Basically, what they did was plant this tree diversity experiment in old agricultural fields that had been in corn for many, many years. And this watershed is actually, this is the Chesapeake Bay. This watershed was actually one of the watersheds where they first determined that riparian buffers help prevent runoff of nitrogen into the bay. So this was actually the ag watershed that they used. And so they replanted it in this diversity experiment with about 18,000 trees. And so John Parker is the PI who was the brains behind all of this, while really also the like mowing muscle in in the future. But he started this along with Dr. Susan Cook Patton, who was a postdoc at the time and is now at the Nature Conservancy. And then this project has tons of people working on it over the years, and I'm going to call them out as I go through. But just know this is kind of a huge effort. So it started with a huge volunteer effort to actually plant the experiment. So some trees in this experiment, and this was planted in 20. So all cornfields, and there are 255 trees that got planted into each plot. The trees that were planted represented a group of species that were the most common species in the surrounding forest. So they span 16 different species, 8 of which are ectomycorrhizal and 8 of which are arbuscular mycorrhizal. All of them are deciduous, and they're all native species. They were planted into plots that either had 1 species, so all in monoculture, had 4 species or 12 species. So those are the 3 different tree couples. And this experiment has been growing ever since then. So the question that I came in when I was starting as a postdoc in 2016 was, what does it matter for insects if a tree is in a monoculture surrounded by all similar things, or if it's surrounded by a diverse set of individual species? And I wanted to look at herbivore abundance and diversity, both kind of by visiting these same trees over and over again and seeing what insects were using them, but also by thinking about kind of the entire community that was being supported, either by plots that were single species or plots that were mixed. And I'll get into this a bit more later, but there's basically different kind of scales that you can think about this at. And so the first piece of the puzzle I'm going to talk about are the caterpillars, because caterpillars are awesome. And when I'm meaning caterpillars here, I'm including both the leaf-feeding larvae of Lepidoptera and the Symphyta sawflies. So functionally, they operate very similarly in our system. They feed on leaves, they act as bird food. From an ecological perspective, they do a lot of the same things. So what I've been doing so the trees were planted in 2013, and I started sampling them in 2016 for caterpillars. So I have been visiting the same 540 trophic trees. We call them trophic trees, since 2016. And each year, kind of, we have a variable number of samples, but each tree has been visited at least once per year, usually twice. The first year, I only sampled the 1 in 12 species plots, but after that, I've been doing all 3 different experiments. And the sampling design is going to be kind of the same for the rest of the evening, and so I'm going to just explain it once now. And that's because these same trees that I'm going to talk about, these same 540 trees, we've been taking all of these measurements on. So it's the same design. And we're really trying to understand how everything is operating in these trees. So basically how it works is each of these different colors is a different species. Here, this is tulip tree, so Liriodendron tulipifera. And so I would take a monoculture of that species. I would pair it with the nearest 4 species and 12 species plots that also contain that same species. So you can see that in this 4 species plot, I would sample 6 trees in the monoculture. I would do this in the neighboring 4 species plot in the same spatial arrangements, and I would do the same thing in the 12 species plot. And what this does is it kind of standardizes the pool of, say, moths and butterflies that are flying over and thinking about whether to oviposit onto these different plots. So they kind of have a choice. And in the experiment, each of these monocultures happens twice. So we have another tulip tree plot, and I've paired it with the 2 nearest 4 and 12 that have it as well. So we did this for 15 of the 16 species in the experiment. And that's how I end up with 540 focal trees. The 16th species in the experiment is a hickory species that took a long time to establish, and we had a lot of mortality. And so there weren't actually enough individuals to sample in the 12 species plots. So I'm going to be talking about 15 species for the rest of this. And so when we go to search for caterpillars, we're actually using visual sampling techniques. We don't want to kind of do destructive techniques that are going to mess with the predators that are on the plots or that are going to potentially alter it in the future, because we want to be able to keep going back to the same trees and see who's colonizing them. And so we did 4 minutes of visual sampling on each tree and basically search vegetation. One thing that's important here is it's not the same people over time, although I am always there. And so for each of the plots in the replicate, we have the same observers. So it's kind of a similar observer bias across each of those replicates, which I think is important for making sure that it's all equivalent. So what are we finding? Well, so far, we've now sampled these 5 different years, and we have about 4,500 caterpillars that we've sampled. Those represent 36 families and about 194 species. And I have Kelsey up here with the caterpillars. She is lab manager in the lab and has really dived into identifying these critters, rearing them, building up collections of images where we can understand what the first instars look like of these species that we're getting. And maybe I'll be able to bring some of them to the also help you with things. But who are we finding? So actually, the most common family that we get are the gracilarids. These are typically leaf mining species, and we get a lot of different critters within that group. We get a lot of tortricids as well. And then we have one softfly group that's quite abundant, the chythrednids, and then actually the only other softfly family we have are the argids. So softflies are a much smaller part of the abundance community here than the Lepidoptera are. But 36 families do show up, which is pretty impressive, I think. So the question here is, well, how does the diversity neighborhood change which critters we've got on these trees? Or does it? Might not. And so when I first started this project as a postdoc, I kind of was trying to predict what I thought would happen. And I wanted to think like an insect. So flipping associational resistance and susceptibility. So associational resistance would be kind of in a group. You might have some kind of buffering where if there are other more resistant plants around you, you might have less things that will attack you. Or on contrast, if you're a non-tasty species, maybe you have lots of tasty things around you, and in a mixed plot, you might get eaten more. So from kind of thinking through all these things, what I ended up predicting was that in our monoculture plots, this concentration of resources would over time end up with more specialist species and perhaps higher abundances. So this is kind of like the resource concentration hypothesis, although not exactly. And then I predicted that I would have fewer insects using the plots in polyculture, but they would maybe have higher diversity and be more generalized. So what did we actually find? Well, this is first, these are the abundance results. And so I just told you I predicted lower abundance on the lower abundance in the diverse plots, and I get the exact opposite result. So here on the x-axis, we're going to have the plot diversity. So this is 1, 4, 12. And then on the x-axis is the caterpillar abundance. And so you can see in 2016, I'm getting I'm getting 87% more caterpillars on trees that are surrounded by diverse neighbors. Does that make sense? Okay. Show you a few of these. Other way. Okay, so we kept sampling, right? And it is extremely consistent. So I was kind of like, okay, I didn't find what my hypothesis thought, but maybe things are going to change. It's not true. This is like a very robust result. I've now looked at this for 5 different years, and we always find more caterpillars on trees when they're surrounded by a diverse mixture, on average. So first step, I was wrong. So this is how this might go today. And so we're not actually finding that we have higher abundances in these monocultures where you have a lot of clumped, similar host plants. So why might this be? Well, maybe there's differences in the plant species densities that are driving some of this. Maybe if you actually have fewer of a type of species that's preferred, and they are they're kind of like restricted to being on those hosts that are like if they wanted to migrate to the next host, it wouldn't be appropriate for them. So maybe they would come back and recolonize that same tree rather than kind of spreading out throughout the plot. So that's a possible. Maybe there are differences in detection of hosts as you're moving over the landscape. I've been tossing around this idea of possibly having some host-associated differentiation driving it. I'd love to talk about any other ideas that folks have for why this might be the case in this experiment, because I'm writing this up right now and reviewers aren't loving this. So what now is happening with diversity? Because I was already really wrong once. So diversity, remember, I predicted would be higher in the diverse plots, and we are seeing this pattern as well. So there's greater abundance and diversity on trees in the diverse plots. And I found this in 2016 when I first started sampling, and that pattern has also been consistent over time. But the slope of this has been decreasing in our most recent samples. And so I'm not 100% sure what's happening there, but I'll be talking more later about kind of what's happening in the plots as the trees are growing, and this could have something to do with this. So take home is that we get more different species using trees very consistently if they're surrounded by other different species. And I'm happy to talk more about all the species we found, but this is just kind of a example of the rank abundance curves for the species that we're getting, either on the monoculture plots are in black, or the 12 species plots are in green. And we basically have the Caleroa is an oak specialist sawfly that is extraordinarily abundant in both monoculture and polyculture plots. But then we have a generalist that's the most common on the monoculture plots, and a specialist that's the second most, or that is the most common on the polyculture plots. So there's a lot to pick apart here, but basically what you can see if you look at the top is that these are on different scales. Actually, you can't see that because there's a block. But basically, the most common species has 400 individuals that we sampled. This is a leaf tire on sycamore. And that same species, we only have gotten 170 of them total across all of our samplings. So there's a pretty large difference in the most abundant species between both of these types of plots. I would say there's differences in community structure. And we have a more even but less abundant structure in monoculture plots compared to the polyculture plots. And I'm going to explain a little bit more about why I think this is. So one of the interesting things here is that we can kind of watch the plots be colonized. So this is Platanus occidentalis sycamore, and each of these are just a collection over time. And these are collector curves for the species diversity. So this is the richness we've gotten after each of our samplings. And what you can see is that we've gotten probably like 26 species of caterpillars that use clock in the diverse plot. We only get like 18 or 19 that use them in the monocultures. So there's just species that are not using the same tree species when it's growing in monoculture versus polyculture. Now, this is our largest species, clock. I will say when we go to the smallest species that we have, this is hickory, we see this pattern reverse, and we actually have greater diversity using the individuals in the monoculture plots. This is unusual, but I wanted to point out that this kind of flip is happening with our smallest and largest plots. If we go and look at all of them, I don't want you to look at all of them, but basically what you can see is for the large species, we really are having these really big differences in diversity that kind of pop up, and then sometimes they go back away, but there's a temporal aspect to colonization that I think we can kind of pick apart a little bit using these collector curves. Okay, so next I was interested in kind of how these tree diversity neighborhoods alter host use patterns across all of these plots. And to do this, we basically built interaction networks. So this is kind of asking the question of whether our interactions are more specialized in monocultures versus polycultures. And so if you haven't seen these, basically the tree species are on the bottom here. Each color is a different tree species. And then everything up here is a different species of caterpillar. And they're connected if there's a feeding relationship documented. So we can build these networks, and then we can calculate matrices like connectance or generality to tell us something about how all of these species are interacting. We can do the same thing for all of the species we found in our 12 species plots. I know you can't read these, that's totally fine. In the 12 species plots, but what I do want you to notice is all of these highlighted species here are species that we only found in that diversity plot. So these are kind of like unique species that we didn't find in the other type of plot. And so there's quite a number of these. And so if you add them all up, we're kind of finding 2 times more unique caterpillars using the 12 species plots. And of the 411 feeding interactions that we documented, there's a greater proportion of them that are kind of unique interactions in the 12 species plots. So this is all very complicated, but we tried to get at this question of specialization by basically using these repeated networks that we had sampled to calculate realized metrics of generality or specialization in the experiment. And so we have an interaction network per sample. So we basically, over this time, have 60 networks that we can look at, monoculture networks, 12 species networks. And here what I'm going to show is the metric of generality. So generality is this idea of what is the average number of plant species a caterpillar species is eating during kind of a given sample. And so this gets at how generalized the feeders are. And so when we look at this across the years of the experiment, what we can basically see is that in general, generality is greater in the 12 species plots. So this means that we find caterpillars feeding on more different species in the network in the mixed plots versus the plots. And this, for example, this saddleback caterpillar, we have found on 3 different species in the 12 species plots. We've only ever found it on red oak in the monoculture plots. So perhaps there's some spillover happening here, where when a tree is growing in amongst other species, some of these species, if they're found, are more likely to kind of jump over and feed on some neighboring plants. But maybe they're not likely to kind of find their host in that mixed plot to begin with. So I don't know, I'm happy to kind of hear how people think about interpreting this result. So in general, what we're finding is the realized feeding breadth is more specialized in the single species plots. So again, I was wrong about abundance, but we are seeing these predictions where we're getting these more generalized feeding networks and higher diversity that are using trees that are surrounded by these diverse habitats. Okay, so any questions about caterpillars? Yeah.
Did you think about how the caterpillar natural enemies might be affected by the more diverse as versus the monocultural, the tritrophic effects.
Yes, and we will get to that with predators.
Oh, I'm sorry.
But,
Okay.
yeah, but that is definitely a good thought. Any like clarification kind of things? Yeah.
How was the tree growth in the 1 species plots versus the 12 species plots?
Okay, we're gonna do that with plants. So, all right, I will keep going, and then we can come through. But you're totally, all of these things are great.
Okay, I have one more legitimate caterpillar question.
Uh-huh.
As the trees grew, did you still sample at the ground level, or did you have any way to reach up into the canopy?
Yes. So all of these we were sampling kind of within where we could reach from the ground, and so that meant that as the trees get bigger, we are sampling kind of a smaller portion of the canopy, but it's similar across the treatments, right? Because the trees, like the hickories, are smaller, but we're doing them across, comparing them just to hickories in the experimental design. So the sycamores got real tall, but we're still sampling the lower vegetation. Now, the reason this only goes to 2022 is that many of these species now have outgrown our ability to sample them with this technique because they've dropped their lower branches, and so we can no longer. So we keep thinking about bucket trucks, but 500 you can't actually get to do these. So I'm gonna have to think creatively about how to continue. Stilts. Yes, I'm sure that like the field safety course that Maryland would have to have us do with stilts. We have to have training for greater than a 3-foot ladder, so what steps to go.
I liked your bipartite network threads, but is there any way that you can assess the component communities for each of the 15 tree species and compare those component communities with each other? Or maybe looking at a bipartite network approach.
So then you would only have one species at the bottom though, so you wouldn't have a network anymore.
Yeah, so I agreed. So you wouldn't have a network. Is there a way that you can actually look at the tissues that are being consumed and then develop a
Okay.
component community based on the tissues, the xylem, epidermis, mesophyll, parenchyma, things like that?
Yeah, so I think that for here we would, so these are all chewing herbivores. So pretty much our sort of functional group differences are miners, tires. We have like etchers, sort of. Yeah, so things that feed on different. I could do that. Yeah.
That could be really illuminating in terms of looking at which tissues are being consumed and the frequency of consumption. One way of looking at that is that on your bipartite network, you can actually look at frequencies, and that can give you interaction strengths of the links between the plants and the herbivores.
Yeah, so the kind of the width of those bars are those weighted.
So we've done that with the fossil record actually.
Cool.
Fong has done this, and it requires a lot of work because you're looking at individual feeding occurrences at the leaf level or at the organ level.
So you think the same species is feeding differently, or would this be like a species level?
It would be both. You could have the same species feeding differently on different plants, or you can have different species feeding similarly on different plants.
Yeah, so we probably couldn't go back in time now in the experiment to relook at new ones, but yeah, that's a great. Let me think about that. That's great. I love it. I love extra ideas. Because this data, I would love to figure out new ways to think about analyzing it. Because it was a lot of work. And,
it is.
A more basic question. When a new tree species comes, it attracts a different species of insect. Where's that insect come from?
Well, I think in this case, those insects are primarily coming from the forest around the experiment. So they probably, all these species are present in that surrounding kind of forest community. So I think that a lot of those are kind of, they're migrating in. But I would say, it's interesting, we've gotten turnover in the species that we're getting. There are some species that when these were all like in the middle of an open field that we got tons of kind of on very young trees. And now some of those species have dropped out of the community entirely. So there does seem to be some specialization, say, on young trees. That's really interesting, and I would love to understand. So I'm kind of going to contrast the caterpillars a little bit to cicadas. And this is the work that Kristen did. And cicadas are really interesting because, like you're saying, all of those lep species and things, they're existing in that community around the plots. The cicadas, this is a brood 10 cicada that came out, and it's a 17-year periodic cicada. So they come out all at once in mass quantities, and they're not this community that sort of builds up over time. And we actually thought that this experiment had no brood cicadas. This is what we were told. And we were planning on using this site as kind of a no cicada comparison for John Little's work, working on cicadas. And then it turns out there were tons of cicadas that came out of this forest. It's just they don't have any entomologists at SERC, and nobody who had been there 17 years ago. And so Kristen sort of took advantage of this and quickly put together an experiment to kind of look at how this mass emergence of cicadas that I will note, none of them were feeding on these trees because the trees were planted in 2013 in cornfields. So the previous time that all of these cicadas were laid, they were laid in the forests. And so you have this mass emergence, and now they have new hosts, basically. And so they have a choice whether to oviposit in the tissue of the monocultures or the 12 species plots. And so Kristen went out to measure this. And so cicadas can feed actually a little bit as adults on plants, but that's probably not how they're primarily doing their damage. Instead, when the cicadas emerge from the ground and then mate, the female then goes and finds places to lay eggs. And this is the ovipositor that's slicing a hole into it. And you can sort of see the abdomen pumping here. And each of those pumps is like another egg going into the plant. And so it's really these slits that can potentially cause damage to the plant. So this is actually Kristen. But here you can see these different like slits on the plant. And what ultimately can happen is that you can get breakage. And then that branch will kind of die further on down past the tree. And that causes damage because it's photosynthetic. So when Kristen went and measured this, basically, she found the opposite pattern to what we found with the caterpillars. So we get more caterpillars using trees that are in diverse plots. This is oviposition scar density. So it's basically how dense those scars are on the tree. And there basically is much lower densities of scars on trees that are growing in 12 species plots for most species. So each of these lines is a different species. And there's also a diversity by species interaction happening here. But it's primarily with a couple of these species where we get a little bit of increased oviposition in the 12 species plots. I think this is associational susceptibility because these are non-preferred species for the most part. But we can discuss that if you're interested. So basically, cicadas are not ovipositing as much in these diverse plots. And that is probably good for the diverse plots because this was a white oak plot after the cicadas came through. So the flagging damage was pretty intensive. And you can see this is white oak. You can see it was one of our species that had the highest densities of scars. And we see this when we go and look at the flagging. So we measured kind of that overall % flagging that was happening at each tree level. And we again see that it's a pretty large decrease in overall flagging that's happening on this trees. So cicadas really don't like these diverse plots. And this kind of got us wondering, how do cicadas pick trees? Kristen and I talked about this out in the fields a lot while we were watching these cicadas flying around to make decisions. And The previous results really show they do care about species, so they are choosing particular species to oviposit on, but they also care about diversity. And we weren't sure kind of why they would care about diversity. And so one idea is that it's light. Like the cicadas are really ovipositing where it's light. Another idea is they might just choose the tallest plant. Or perhaps they like trees that are really rapidly growing because then they have lots of new tissue to oviposit in, right? And so we've been trying to figure this out by using some path analysis models. The nice part about this experiment, again, is I've been visiting these trees for lots of years now, probably more years than some people think is a good idea. And so we actually know how quickly the trees were growing the year before the cicadas showed up. And we know what their heights were the year that the cicadas happened, and we know what their canopy density was the summer that the cicadas emerged. And so we basically wanted to run this path analysis to ask, is diversity affecting any of these things in a way that changes how cicadas choose? And so what we ended up finding, so these are all of the grayed out paths or paths we hypothesized might be the case, but turned out not to be important. What it really seems to be is that diverse plots have trees that have more dense canopies. And so these black arrows are a positive relationship. So richness increases focal tree canopy diversity. Height also does this. I mean, this is logical. If you have a taller tree and you're looking at the canopy cover, it's going to be greater. But it's really that canopy density that is decreasing cicada oviposition. So these trees have these more dense canopy, and then you get this decreased scar density. Interestingly, if they're growing fast before the emergence, that also has a positive effect on cicada oviposition. So it doesn't seem like they care that much about tree height, but they care about whether the tree was growing fast, and they care about how sort of light it is. And so then we wanted to ask, okay, well, does this oviposition actually lead to that flagging damage that we see? And we can add that to this model, and basically we can see that if you were growing faster, you flag more. That kind of makes sense. Your wood is going to be weaker, perhaps. But also, we get if you have more scars, you flag more. So we can show that that's happening. And then, actually, we had a negative impact of this flagging on the height growth of that tree the year the cicadas emerge. So we can see that the cicadas are having an immediate small effect on relative height rate growth of these trees, probably because they're causing this flagging damage. But does this kind of does this affect things moving forward? And it doesn't seem like it is. So we've continued measuring these trees for the last 2 years. And basically, trees that had more flagging actually are growing. They're expanding their root collars more in the last 2 years than trees that didn't have flagging. So this could be perhaps due to a pruning effect, maybe, where they're kind of increasing their girth because they got kind of pruned down. They also could have a lot of cicada carcasses around them, so it could be fertilization that's driving this. We don't really know exactly what's happening here. But the interesting thing is that the cicadas really don't seem to be having a lasting negative impact on these trees at all, which is kind of cool. Okay, so for cicadas, actually we get way more of them in monoculture plots than diverse plots. And so why might this be different from our caterpillars? It could be that this outbreak event is very different. You have a high density emergence where they're in direct kind of competition for space with one another. Maybe there's an aggregation aspect for these species. I would really like to, count the emergence holes from these plots in like 17 years.
No, we have to wait 15 more.
15 years, yeah, until these guys come out again to see whether this actually is having an effect on the root feeding, right? Because if we had less cicada oviposition happening during this mass event, you would expect fewer babies to fall down to colonize the roots of each of these trees. So our experiment now is cicadas and below ground herbivory, whether we wanted it to or not, for the foreseeable future. And so I think what this is starting to point to is that really, insects are living in this environment that's created by tree-tree interactions. And so we're looking at these responses of insects, but really they are thinking about what the plants are doing. At least that's what the cicadas are telling us. And so we have been measuring traits a little bit, but we really did a lot of measurements in 2021. And what we can see is the tree traits are different in the plots of different diversities. So things like leaf dry matter content, which we think about, how, and leaf mass area, these are metrics of kind of how much investment you're putting into your leaves, toughness, things like that. These are all lower in our diverse plots, and relative chlorophyll content is higher. This is non-photosynthetic quenching. This is a measure of kind of the ability of leaves to dissipate excess heat during photosynthesis, and that is also lower in these diverse plots. And leaf temperature differential is lower. So really what this is telling you is we have shade leaves in our diverse plots compared to the monoculture plots. And we see this with the tree canopy cover too, like I mentioned. So these insects are really responding to the tree interactions that are happening. And so what is happening with those? So this is work that was done by a postdoc in the lab who's left and got a job at Kenyon. And this is work kind of trying to understand how these trees are actually growing. And we didn't want to just look at height or just look at basal area. We wanted to understand more about the size and architecture of these individual trees. And so we've been measuring these annually during the dormant season, and we calculated basal area and canopy volume and are looking at it over time. And so what we're finding is that we are getting larger trees in terms of height in diverse plots, but only by a little bit. So this dotted line is the monoculture, and then 4 species and 12 species. When we look at basal area, though, this is where we're starting to see a pretty big difference, and a difference that's increasing over time between our different plots. And when we look at canopy volume, we're getting to even larger numbers. And I'll explain what this means in a second. So over time, we're getting these increasing differences. And so what Lauren found is that basically the large trees in these polyculture plots look very different, and they have different architectures than the large trees in the monoculture plots. This is height on the x-axis and then the basal area here. And so basically, as you get taller, how much basal area you're having, or how much canopy volume are you having. And so what you can see is that in the monoculture plots, you basically don't increase your canopy volume as you get taller. Whereas in these 12 species plots, you're getting much wider as you get taller. And so we're getting these kind of large wide trees versus narrow trees. And this makes sense when we think about kind of crown plasticity and vertical stratification in these plots. So the sycamores are one of our fastest growing species, and they basically shot up. They're now about 17 meters tall after 10 or 11 years. Very, very quick growth. And they grew, like the monocultures grew much faster than the polycultures in terms of height, because they're competing with each other to reach that light. And so we end up with these tall trees, but then over time they lose their canopy because they're all competing with each other at the same height. So they lost their branches very quickly, the lower branches. In contrast, in our 12 species plots, we have trees that are kind of at different heights. And so you get this canopy packing where you get kind of larger individual trees that are able to fit into space and can all gain light. Does that make sense? So here you can kind of see this. This is the sycamore plots. This is John. And so they basically, the ones in the center here are like a lollipop with just sort of like branches at the top, whereas the ones in the mixed plots are much wider. And so we end up with kind of these different habitats that the caterpillars say that we sampled before are living in, either in the sun or in these really dense shade conditions. And so what does this mean for predators? I don't have any tachinid data or parasite data, but here are some tachinids that are on this guy. It's simple. So Elizabeth Butts looked at this with spiders in the plots in 2021. And she did indeed find, as was suggested, that we are getting more spiders that are using the diverse plots. So if we go to those same trees, we look at how many spiders are there, there's more spiders. And the reason, again, is that the spiders like trees with more canopy cover. So we did some path analysis here, and basically treatment is not actually affecting, is affecting our spider densities, but it's doing so by changing canopy cover and not by changing height. And interestingly enough, treatment does increase our focal prey density of caterpillars, but that is not what explains spiders. Spiders really are kind of selecting these trees that have more canopy cover, maybe for, this is in August, late summer, it's very hot. So perhaps they're looking for microclimate protection within these plots. Or perhaps they have more prey items across the whole season as well. All right, so those are all kind of the vignettes here. And I've been working on this for a while, and this was sort of nice to step back and try to understand multiple different pieces that we're sort of trying to put the puzzle together on. And so overall, what I've shown you is that basically caterpillars really do better on trees that are growing in diverse plots in terms of diversity and abundance. But you get a lower realized host utilization in those plots. You get trees that are stouter, have more tender leaves, and have more canopy cover in these diverse plots. And you get lower cicada oviposition and immediate damage in diverse plots. And we get more spiders. So kind of thinking about this from an ecological perspective, I would say that probably you have more like robust food webs that are happening in these more diverse plots. And this is important because there's lots of critters that rely on all of our insects. These are sawflies actually, but the caterpillars. And there's lots of ecosystem services these guys provide. And we don't know what's happening with some of the other predators out there. Kristen might have some information on parasitization rates of some of these caterpillars at some point, but that could be another way that we're seeing kind of these differences. So I think that what this experiment is kind of getting at is that diversity can create an increased robustness in our plant insect interaction networks that might help play a role in potentially doing things like moderating pest outbreaks in more managed systems like parks. We didn't actually have any pest outbreaks in this experiment, probably because it's in a more forested type ecosystem. But if you are able to support more predators and kind of more alternative prey items over the season, you probably will have fewer pest outbreaks. An interesting part of this is that it does alter the biophysical property of stands. And you can also think about this in more human-modified landscapes as well, when we're thinking about trying to create urban cooling, for example. Maybe having different layers of vegetation can help with some of those things rather than having everything the same place. And again, USDA is investing planting trees everywhere. So I think that it's important to kind of think about how we're going to do those plantings. I've been working with a number of folks in Baltimore and the urban tree program. I'm trying to get increased diversity within kind of planting plants for that to pest outbreaks. I think that's something that as entomologists, we can all sort of do is encourage people to create plantings that are more diverse because often I feel like I'm called in once sort of there's already a problem because, somebody planted a whole line of these are ash, and this is actually, before emerald ash borer. This is clearwing, banded ash clearwing damage. But if you kind of change the arrangement of trees on the landscape, that has implications for which insects are in those. Cool. So after 10 years, I would say the experiment's very cool. I encourage anybody, if you have an insect group that you're interested in, like talk to John. He's very interested in having people sample out there. I want to know about the galls. The synipid oak galls are really coming in force now, and I really want to sample them, but I haven't been able to get in return. Kristen's heard me talking about this for like 30 years. I can't get anybody interested in the galls. Yeah, there's a lot of work involved with this, but it also takes a really long-term investment. I think Smithsonian is really a great place to do experiments like this. It takes a long time, but also like nothing is standing still in the experiment. Like everything is growing. And so you can never go and measure the same forest twice. And I think that sounds trite, but like working in this for a while, it feels actually very true. So I think there's a lot of direct conservation and policy implications here. And this is Kristen. Their plots. This is actually not me. This is my twin sister who came out and helped. So it's a lot of work. We even have to get our siblings to help. And so where are we moving forward with this? We have an NSF grant to try to connect what I show you today with like insect herbivores and leaf traits to the tree microbiome. And so there's a whole bunch of folks who are part of this project. We sampled all of this simultaneously on the same trees we've been tracking in 2022 for all 15 species and are working on all that data. We have some back, but this is just an example of the 2017 data where we're trying to do path analyses for these. And in 2017, we're trying to make connections between caterpillars and the phytochemical diversity in these leaves. So is diversity changing something about the expression of chemistry in the leaf that then is changing kind of how these food webs operate? And so these are our hypothesized kind of relationships here, where maybe tree richness is going to increase phytochemical diversity, which will decrease our caterpillars and decrease deer damage and end up kind of having a positive impact on growth. And what are we finding? So this is our data from 2017 from the same trees. And basically here, we have tree richness having this positive effect on phytochemical diversity in the leaves, actually through heterocyclics. I'll be talking about that. But that diversity has a negative impact on deer damage. So the deer seem to care about that. And ultimately, it's the deer that are really having a negative impact on tree growth. This is in 2017. So the trees were small. This is 3 years after planting. So deer were huge in the system back then. Interestingly for me, tree richness is increasing caterpillars and phytochemical diversity is increasing caterpillars, but actually, and increasing damage, but that is not having a negative impact on tree growth. It's actually a positive impact on tree growth. And what I think is that this is really kind of flipped, and probably the caterpillars are selecting trees that are doing well. And so it's just you're having kind of more caterpillars and things on these trees that are growing well. So I would say that what this is telling us is kind of the caterpillars and insects are along for the ride. And they're not really having major impacts on the tree growth at this point in the experiment. And it's really deer that are kind of driving the bus, at least at this point. So what's going to happen in 5 years on? We don't know. Deer are definitely not having a large impact anymore. Okay, so with that, I'm going to finish up. I'm just showing you another critter right now. This is a caterpillar that fell down onto my apple, was crawling around. And does anybody know what this guy is?
Something skipper.
No, actually. Oh, I'm glad that I trained people up. It's a prominent, actually. It's a Georgian prominent. So it was crawling around. It actually has these really cool polka dotted legs, which like I don't see on many caterpillars at all. So it was going around, and I have 3 oaks in my canopy on my house. Like I have a post oak, a willow oak, and a white oak. So I didn't know what it fed to like put it back on a tree. So I gave it a buffet of kind of different leaves to feed on. And I came back and like I couldn't find it. And I looked for this caterpillar for so long in this dish. I finally found it. It chose willow oak. And it's using this line that's on the side to sort of blend in and mask its damage, right? Very impressive critter. But kind of when I thinking about this experiment and science in general, I feel like lots of the folks who are doing all this work sort of blend in when we do these presentations, but really there's a huge number of folks that are really kind of contributing to all of this. And so I'd like to thank them, and I'm happy to take any questions. Sorry, I didn't have a clock, so yeah.
So this may be an overly broad question and it may be unanswered, but through the ontogeny of this experiment, did you see any evidence or do you suspect that this system started out resource driven from the bottom up, and then it ended up, and you mentioned predators, from a greater occurrence of parasitoids and predators from top down driven system, that in which there may be the herbivores may have been controlled towards the end of the project by top down controls by predators and parasitoids.
Yeah, you know, it's possible. You mean that kind of the shift that we're having a little bit in the diversity over time?
Yeah.
Yeah. You know, it's really hard to get that parasitoid data. So we reared for a couple of years in 2019 and 2021. And we have about 25% parasitization that we're getting. You know, we also only get like 30% adults. So what's happening with that middle? They could be parasitized, they could not. This is a lot of species to try to rear and get. So I would love to have done the rearing over a longer period of time. I suspect that the parasitoids take longer. And also the predators, like the spiders and things, probably have taken longer to kind of accumulate in the plots.
To get established.
Yeah, to get established. And I think that particularly things like the spiders, they are migrating from kind of the surrounding forest often. So into the fields. And then now I think our experiment is the forest. And so in some of the plots, I think they're staying, and we actually just have populations that are there year round now in the closed canopy plots. And then there's still a few species, bizarrely, red maple is one of them, that the plants are still this big because the deer chomp them every year. So the deer are still affecting the red maple. They also chomp the black gum and the elm are two. Those are like kind of our three most preferred species. Interestingly, the oaks, the deer have had very little impact. So I think it's going to be tricky to disentangle those things. I totally think that is a lovely idea, and I would like to be able to test it. I think that now the trees are getting light limited. I think that is what. And so there's kind of, we're in this next canopy closure stage, and I'm really curious to see what's going to happen with the trees then. We sort of have a second wave happening where now the small species that were in monoculture are kind of catching up. So they're hitting that sort of phase where now they're growing big enough to start becoming lollipop trees and are like competing. And so I think that's going to be an interesting thing to watch. But yeah, the species, they don't stand still. Each species is kind of doing its own thing. And this is it's really complicated to try to disentangle all these patterns out, which is probably why it's taking so long. So, any other questions? Yeah.
I wanted to know the way how you collect all the data, because you just said you use different students help you to observe those leaves. But I want to know more detail, like how you get those damage and you record it in the data. Because you said you count the leaf miner also, but you count the leaf chewer also. So how many kind of those data you use inside?
So we did remove all of the caterpillars from the tree that we found. So in that kind of 4-minute sampling window, we destructively harvested. So we would stop, if we found caterpillars, we'd stop the timer, collect them, put them in an individual bag with a label, and then kind of start the timer again to sample. So we collected all the leaf miners, all of the individual brewers, all of those. And then we brought them back to the lab and we would process them there. For the kind of easy to identify, but sort of rare or threatened species, I would actually take the caterpillars and I release them in the forest on the same place of plant. Some of like the Saturnians and things that it's really easy to tell what a polyphenol spot is. So yes, so we would collect all of them, we'd bring them back to the lab. We would take photos in the lab, so there's processing in the lab. So we would get, for the mines, we would take kind of like photos where you could see through the mines. And then we would have photo vouchers, we put them in ethanol. Some of the ones that we couldn't identify, we would send for barcoding. Some of the ones we would try to rear. So I feel like it's been kind of like a puzzle over time of trying to put together what this community is. And we couldn't barcode all 4,500 because that's just like way too expensive. But many of these species, we have good images now of what they look like in all of their instars. And we're trying to like put that together as a resource as well.
You show that line of ashes, and that's a monoculture, but it's also very different than forested plots you had in terms of microclimate. In fact, it almost looks like two aspects of the monoculture that you pointed out are emphasized. Towards you have these literally a monospecies, but you also have what you described, I think, is a much more open and light area, not shaded. So to what extent did then that comes into recommendations like you get this narrow little strip in a parking lot? How can we change the structure of some of our urban environments to get some more benefits, not just even if we have a diverse species?
Yeah, so other kind of work in the lab, first, yes, I think planting a diverse kind of group can help you because with a lot of these pest species, they can kind of walk down a line of trees, right? So if you have sort of, you can see this really well with like oak stem gall. If you've ever seen an outbreak of that, you'll often find it in sort of like a line of getting more severe outbreak because basically they've jumped from trees. And then anyway, so I think that the diversity piece can help. I think that you can create larger places for the trees to put their roots down. So a lot of the problem also is impervious surface and heat. That is really fairly negative for a lot of the predators and other critters that are going to kind of help you out in terms of biocontrol in those spaces. I mean, I think a lot of our other management tactics, like removing all of the leaf litter, for example, is one thing that can decrease our potential to have natural enemies and biological control happening in those types of settings. If the predators have to find and colonize your trees every year, you're never going to have them be in abundances that are going to be able to control your potential pest species. And then we also mulch in ways that are really like detrimental to a lot of the trees. On my walk to work, I pass by these maples that have mulch like this far up the stack, and they have borers that are coming out of them. They're dying, and so there's cultural techniques that really can actually help these trees be less stressed to begin with. And then if you do get kind of these insects on them, it's not as big of a deal, right? Which I think is what's happening in this experiment. The insects just aren't that big of a deal because the trees are kind of generally doing okay. And there's lots of predators. The birds, I think, are a huge part of this as well. They've increased a lot in the experiment. We did some plasticine caterpillar studies in 2017 and didn't see big differences, but I kind of think now that it's tricky because I don't want to put plasticine caterpillars on these trees because then I'm going to attract birds, and then I won't get the caterpillar measurements, right? Because they're just gonna. So anyway, it's tricky when you're kind of trying not to mess with the experiment too much, but yeah, I don't know. I mean, I think there's lots of things we can do at the planting stage and the design stage that would make these urban landscapes a lot more hospitable to a lot of creatures that would help you prevent pests.
And then, of course, there's the question of
how do we remake some of the, not so advantageous urban landscapes we have?
I mean, so a lot of what they're doing in Baltimore is actually like site rehab with these tree pits and things. So actually going in, taking out all of the soil, widening it, making it a lot bigger. And that actually is how you're going to have a successful tree growing there. A lot of the situations where we try to just put these trees into areas that have fill or like just are not kind of prepped properly, that's just going to be a failing task. And this is one of the reasons probably we have a lot of inequities in our tree cover across cities. And so some of the work that our lab's been doing was looking at street tree censuses in Baltimore, and areas that have been previously redlined back in like the 1940s. They have lower tree cover now, but also the tree species that are being planted in them are less diverse. And so you can get these feedback loops where probably they're going to die more because they're less diverse and pests can potentially attack them. There, they actually plant a lot of red maple. Red maple is just coming up a lot today. That in some neighborhoods, it's like 20% of the trees that are being planted, 25%. And I don't know, we know Asian longhorn beetle will love it if it ever gets established. It's tricky. I mean, urban landscapes are really tricky to figure out, but I think we can get some lessons from experiments.
So I'm kind of curious, do you think that what you found with caterpillars extrapolates out to other taxa? Like I'm thinking beetles and flies. Like anecdotally, when you're walking around underneath of, let's say the more diverse plot, do you notice that it's like super buggy? Are you getting eaten alive when you're under there? Or is it, versus the one tree plot?
Yeah, I think it's tricky. So I will say that for this study, the 2022 to 2024 one that we're doing, we are looking at hemipterans as well in the experiment, because we have this idea that maybe some of like the leaf endophytes and pathogens are getting spread around by some of the hemipteran communities, because they do a lot more kind of running and jumping around than the caterpillars do. I mean, we do have some ballooners and things, but I don't think the caterpillars are probably transmitting pathogens amongst individual trees, whereas I do think that hemipterans could. And we haven't analyzed that data yet. Anecdotally, I think that for a lot of the generalist species that we're getting, so the sharpshooters and things like that, I'm not seeing a difference. And we're not going to be able to get to species with that because we aren't actually collecting them. So it's just a family, like visual family kind of stuff. So
when you guys were doing the breakdown by family for the abundance, did you see any major differences? Like, I don't know what kind of damage I don't know anything about caterpillars, but if there are different kinds of, if they have different like ecological functions, did you see any differences there?
So I think that the gracilarids that are a lot of leaf liners, I think we are getting more of those in the spots. Yeah, I don't know that we've made a pie chart for each of the different diversity treatments. That is a great idea. This is why it's good to talk to people.
Do you have any questions from the virtual audience? Well, if there are no more questions,
There might be one from Alan.
Yeah.
You mentioned like wanting to study the old calls. Did you put out any feelers on that galling listserv?
There's a galling listserv. I don't. I mean, this is a great name for a listserv.
It's a lot of gall.
Yeah. Yeah.
Somebody at Penn State.
Oh yeah. So okay. Yeah, so I was talking with John Tooker about this a little bit. He's working with, I'm blanking on his name, from Penn State, who I know they've been looking for the sexual generation of that jumping oak ball. Do you know what I'm talking about? It's like a Neurotaurus species that we don't know what it is. Some of them have white oaks and they have those little galls. They look like, I don't know. little bubbles and they fall off in large quantities. They're super tiny. I have them all over my house. Like it will literally be millions. Like they cover, and so this is a newish species, maybe. There's this jumping gall wasp, Neurotorus sultoris, which is from the West, California, and it like jumps. These don't jump. So he's trying to figure out which species this actually is. So yes, but I feel like I could ask, maybe I need to recruit people specifically for the gall thing. I feel like I have people interview for positions in the lab, and then I try to like talk to them about projects, and I don't know, I can't get, the galls are so cool, like, anyway.
So that's just questions I don't really follow as much.
Yeah.
It's all different things.
Well, they can be really complicated. I think here it's sort of a manageable number of sort of morphological types of galls. And we probably would only do
You can also ask Mac Buffington.
He was on my list to talk to, but I don't think he's here tonight.
No.
So I'll send him an email. Cool.
Letters and comments.
Well, that's a fun time of life.
Okay, thank you very much, Karin.
And working.
No objections. Meeting is adjourned.